Hello, I, Santoshi N. Krishnan, along with my teammates Meghna Hukkeri and C.R. Parvati, are currently pursuing our fourth year in biomedical engineering at SRM University, Katankalatu. Our fourth year project is entitled Multi-Parameter Analysis of Human Emotional States, and this was done under the guidance of Dr. M. Amburajan, who is the professor and head of the biomedical engineering department. In our project, we aim to evaluate a classification pattern for different emotional states based on real-time simultaneous monitoring of three physiological signals: facial skin temperature, heart rate, and galvanic skin resistance in normal young adults. This is done by fulfilling four main objectives. First, to construct an experimental setup using suitable sensors, biomedical workbench, and infrared thermal camera to simultaneously acquire and store galvanic skin resistance, heart rate, and dynamic phase thermogram of the young adult individual when they observe a set of standard pictures of different emotions studied. Next, to measure the average facial temperature at forehead, periorbital, nose and mouth region of the dynamic phase thermogram using software. Then, to perform statistical analysis of the parameters obtained in time domain after normalization of the values for subject independence. Finally, to evaluate the changes in the parameters studied with respect to different emotions of the young adult individual.
This experiment is held in the Biomedical Instrumentation Lab at the Department of Biomedical Engineering, SRM University. As you can see, this is the setup of the experiment with the subject based at that position and the recorder who is recording thermal imaging. Now the subject is isolated from the surroundings through wooden boards to provide vision isolation, and auditory isolation is provided using headphones through which we provide white noise to that person. Now the distance between the thermal camera and the person is maintained to be 100 centimeters or 1 meter, in front of which we have placed the laptop in which we show the images to induce emotions onto the subject. On the side of the subject are the two sensors to which he is connected, along with the interfacing through NILVIS as well as the LabVIEW program, which is doing the acquisition and storage, while another laptop controls the FLIR module through which acquisition of dynamic thermal video is done. As you can see, this is how the value is being acquired, and it is stored in an Excel file. And this is the interfacing module where the sensor is connected, and the blink indicates the heartbeat at that particular point of time. Now this is basically the GAPAD database that we have used for the induction of emotions. In this case, as you can see, we are inducing the emotion of anger onto the subject. Now this is basically the thermal acquisition module where a person sitting, the second person sitting will control the thermal acquisition taking place using FLIR Research IR software. For the purpose of interfacing a pulse oximetry sensor as well as a galvanic skin resistance sensor, we use the NI-LVIS2 board that is manufactured by National Instruments. Now coming to the specific connections, we use AI0 input analog input pins, positive and negative, for connecting your pulse oximetry sensor, and we use the positive and negative pins of AI7 to connect our galvanic skin resistance sensor. Now for showing the output that the NI-LVIS has taken on LabVIEW, what we do is we use a tool called as DAC Assistant. Now in DAC Assistant, if you go to properties, you will be able to select the channels. That for voltage 0 over here is already pre-selected for AI0, and for the second one is selected as AI7. After the channels are selected, we decide the maximum and minimum, which is specified by the manufacturers. After that, we select the thermal configurations to let NIDAC choose. We check if the samples to read and the rate is as specified, and we click OK. Once this is done, and the VI is built, we run the program to display the output on 3 waveform graphs. The first one showing the combined waveform and the other two showing them individually. Now for this purpose of storing the values for an analysis, we use the write to spreadsheet command, which for both the values separately, and we've created 2 separate Excel sheets for both of them, which can be specified over here, and then they're stored for further use. For the purpose of acquisition and analysis of a dynamic thermal video, we use the FLIR ResearchIR software, which is interfaced with your infrared camera. Now in this case, what we've shown is already a pre-recorded video of a person who has anger induced onto him. Now when you play this video, we have already taken into account the time duration before and after. We first select plot, then we select average, and we add the plot window, which gives you the value throughout the duration of the video. We do similarly for the other 2 regions of interest as well. And we get the plot for the 3 different values color coded by the color that's used in the image. Now the 3 regions that we have chosen in this case is the forehead region, then we have chosen the periorbital region, and we've chosen the nose and mouth region, the 3 regions susceptible to changes in temperature. Now once this is obtained, what we do is to save the specific values for each time duration. We go on to export and we click on data to CSV. There we take the time. In this case we can choose anger subject 5 because it is the 5th subject, and we save it. So in this way, what we see is that the average values for each small duration of time is saved onto an Excel sheet for further analysis.
From our study, it can be concluded that from all the 3 parameters measured, the heart rate showed the most significant changes in value when observed with respect to the neutral state, when there is a comparison done between the positive and negative valence states, and when the negative valence emotions were compared to each other. This is reflected more so in the negative valence states. Facial dynamic imaging, on the other hand, showed the highest overall regional sensitivity. With reference to the neutral state, the forehead temperature showed the highest established significant difference in study, while for intra-valency and cross-valency comparisons, the forehead and the oronasal region respectively were the best judges for change in emotions, and thus the best parameter to be studied when a classified system of emotions is